All right, so this video is on ribosomes. There we go, as soon as it'll load. And so ribosomes, their function is protein synthesis. So they are going to be doing the process of translation. They're going to be receiving the mRNA that's coming from the nucleus that just came from transcription. And they're going to bind to that mRNA. They're going to read it one base at a time, or 3 bases at a time, a codon at a time, and bring in the correct tRNA to build that amino acid chain. We'll talk way more about that in Unit 6, I believe, but just kind of a refresher. So it is made of rRNA and protein. I talked a little bit about that in the nucleus video because they are assembled in the nucleolus. And so we talked a little bit about how that happens. And all cells have ribosomes. That is not true for all organelles. Your membrane-bound organelles, like your nucleus, mitochondria, chloroplasts, Golgi, all those, that are either made of membranes or surrounded by a membrane are not found in prokaryotes. They're only found in eukaryotes. But ribosomes are not membrane bound, and they are found in all cells. So prokaryotes have them, eukaryotes have them. They are slightly different in prokaryotes and eukaryotes, and we'll talk about that, or we'll talk about that next week with the endosymbiosis theory a little bit. But they still both have ribosomes, and they are similar. So there are two types, I guess you could say, of ribosomes, or two places you would find ribosomes. The first are what we call free ribosomes, and they are just floating around in the cytosol. So that would be this number 3 on here. So they're just kind of hanging out, and they're generally making proteins that are going to stay within the cell. And so they're going to make a protein, and that protein is going to get shipped to the mitochondria or to the lysosome or to the nucleus or whatever. On the other hand, there are bound ribosomes, and they are bound to the rough ER, which is why it's called the rough ER, because it has all these little ribosomes all over it. And so that would be these here. And they are generally going to make proteins that are going to leave the cell, because they're going to send them through the rough ER to the Golgi, and the Golgi will send them to the cell membrane for exocytosis. So generally speaking, your proteins made in the bound ribosomes are leaving the cell. The ones made by the free ribosomes are staying in the cell. So what does a ribosome look like? Well, when you zoom in, a ribosome is made of 2 subunits, a large subunit and a small subunit. Real original names there. And these are kind of like coworkers that you don't maybe like to hang out with. They only come together to work. So you are not going to see them in this formation unless it is time for translation. When they're just resting in between translation attempts, they're going to be separated, just doing their own thing. But when it is time, when an mRNA comes out of the nucleus, okay, so it's going to come out of one of these pores out into the cytosol, the 2 subunits are going to come together, and they're going to clamp around that mRNA. The mRNA is here in red. They're going to clamp around it. There's a binding site on the small subunit, and that's going to hold the mRNA in place. Now, there are 3 sites within the ribosome. We call them the APE sites, so it spells ape if you read backwards. The A is where it's the mRNA codon that's being read at the time. So if it's AAA, it would be in this location. And so that's the aminoacyl site, because it's the site that's going to read that codon on the mRNA, and it's going to call in the tRNA with the amino acid on it. We'll talk way more about this in Unit 6. The second site is the peptidyl site. This is where they're actually forming that peptide bond between the 2 amino acids. So this one already has an amino acid. It's already been read. The amino acid's already been brought in. It's got the growing polypeptide chain. You can see it's attached to the tRNA at this P site. And then we have the exit site. So that's just where it's going to set things to go. So structurally, binding site allows it to hold the mRNA in place. The APE sites allow it to read, attach, and then remove as it's building that protein. And so that is how the structure of the ribosome matches its function. I hope that helps.